So let's talk next about stem cells and cloning. We remember this slide from our earlier presentations done some weeks ago. And really, the take-home message here is this time that every cell in your body carries a copy of all genes. So basically, in theory, every cell in your body could become a fully new organism that is you. And this actually happens especially early here in the development, when cells have not yet been defined to have a destiny to become this or that type of tissue. But later on, when we are older, cells typically have become some specific type of tissue. But in theory, they carry all the information that is needed to become any type of cell at all. And this is what cloning uses as its fundamental principle. So there are two types of cloning, reproductive and therapeutic. And we will start by looking at reproductive cloning. And this is something that I am especially interested in. And this story takes us to talk about something that actually some of my friends were involved in. We are going to talk about Dolly the sheep. Dolly was a domesticated sheep. So why is Dolly important for us when we talk about the material in this chapter? And the reason is that Dolly was the first mammal that has been ever cloned from an adult somatic cell. And the big thing here is that this was done from the DNA contained in a mature adult cell. Not what you see here as the early dividing cells of the new individual, but rather these normal mammary cells. And actually, this work was done in Scotland at the Roslin Institute, where a few of my friends work. So Dolly was created artificially by taking an adult cell nucleus and placing it inside a donor cell that had its nucleus removed. And this egg cell was then implanted into a third sheep. So I guess that you could say that Dolly had in fact three mothers. One that provided the egg, another one that gave the DNA, and the third one that carried the embryo to the term. So, how does this technique work? Well, as I said, we take the cell nucleus from an adult cell and transfer it into an unfertilized egg cell that had its DNA removed from, and then once it started to divide, it was implanted into a surrogate. This is something known as a process of nuclear transfer. And there are a few complications, including how to get the egg to divide, and this includes using a kind of an electric shock. But really, this is a very difficult process, and in fact often results in an abnormal development. Actually, it took some 277 attempts before this worked for what was to become a dolly. So it really is a costly and quite inefficient method due to a high failure rate. And I am almost certain that although we hear very little about it in news because no one wants to tell about their failures, this has been attempted for humans too. We have later done many large mammals, including pigs, deer, horses, bulls, and so on. So, why to do this? Well, actually, this could be a solution to protecting endangered species, or even bringing back extinct species. In fact, I think that this has been done in Spain for a local mountain goat that went extinct there for a while. So, let's talk about what happened to Dolly. What is a normal average life expectancy of a sheep? Well, that would be about 11 to 12 years. Dolly lived to 7 years old. So not quite the full life, but still a reasonably long life. And actually, Dolly died of a lung disease. In my view, it probably was good. She had suffered from arthritis and quite a few other medical conditions. The last I heard from my friends, that was in 2016, there were still 4 identical clones to Dolly living there in Scotland, and they were happy and healthy. And I guess that the last question is that why Dolly was named Dolly. And this is a true story. Well, what if I tell you that Dolly's genetic material was derived from an adult mammary gland cell? And when the researchers were thinking of a name for the sheep that they had cloned, the first of its kind, they could not think of a more impressive pair than Dolly Parton. Well, you take that information and do whatever you wish with that. Well, let's go back to our lecture material. I do want to mention, though, that cloning as such is not a totally out there and new idea. For centuries, we have commonly bred farm animals with desirable traits, and do remember that in the US, federally funded research cannot be involved with any experiments involving cloning humans. You might want to remember that it was so difficult to even succeed with Dolly, so this is really a hugely ineffective method. And this reproductive cloning approach is constantly used in plants. That is what we do and have done for a while. Think of cuttings to start a new plant. Here is a fun fact. For example, all the banana trees that we currently have are so much of a clones of each other that there is a real risk of an infection that it is not resistant to appearing spontaneously and wiping them all off. So, a few words about therapeutic cloning. Here we end up with a mature cells that serve some purpose, for example, for treatment of a human illness. And this is done using stem cells. And in adults, most cells are locked in a path of what they can become, so stem cells are not that numerous in their availability. But embryonic cells would be ideal, as they can become anything at all. This work is largely limited in humans by the ethical issues that we have. And here is a diagram showing therapeutic cloning pathway, where these unspecialized cells can become any type that we may need. Well, the next section on biotechnology products I will leave for you to study by yourself. Just remember that transgenic refers to an organism that has foreign gene inserted into it, just like that recombinant DNA method that we talked about. And there are many applications for these methods, some of which we have already mentioned, and these can be in bacteria, plants, or animals.